So this lecture will cover Chapter 8, Precipitation and Particle Agglutination Methods. Now, these first few slides do list several different terms that you should be familiar with. So just make sure that you review each of these slides and review the terms and the definitions, and just make sure that you understand them as we're going into this lecture. Precipitation and agglutination is considered to be unlabeled immunoassays. And so basically the result is looking for the presence of that agglutination or aggregation of those antigens and antibodies to actually form and show that visual aggregation, versus where your labeled immunoassays are basically your antigen or antibody is tagged with something that's going to allow it to be detected in a different way. So this could be something like it being able to emit some sort of radiation, produce a color change, able to fluoresce or emit some sort of light signal. So with the unlabeled, you're just looking for that clumping, agglutination, precipitation that's taken place. Precipitation is the term for the aggregation of soluble test antigens. Precipitation is a combination of soluble antigen with soluble antibody in order to produce a visible insoluble complex. Agglutination is the process whereby specific antigens, so these could be, for example, present on red blood cells, will actually aggregate to form a larger visible clump when the corresponding specific antibody is actually present in the serum. So we do have a depiction here of a couple of different images to kind of try to help explain this just a little bit. So with your precipitation, again, you do have those soluble antigens as well as those soluble antibodies that are in that sample, and then they're going to come together. So you see here where this precipitation reaction here in the center kind of demonstrates how those come together and start kind of bridging together and then forming a particle that's actually visible. So you start off with your suspension, and then depending on the test conditions, whether it's an incubation period, or any other factors that may take place, then you're actually allowing time for those antigens and antibodies to come together. And then what you're looking for is some type of precipitate where they just kind of come together. If you've ever had milk kind of spoil in the refrigerator, and you see how over time those particles kind of start to aggregate together, and then you kind of have this separation that takes place, that kind of gives you the visual of what's happening here. Now with agglutination, you have those antibodies, and then you have those red cells that are present. So you actually can visually see those red cells, but they're kind of dispersed throughout that solution. But once you have that binding actually start to take place, then you're going to actually get clumps of cells that will be more visual. You can see that clumping actually taking place with those cells forming those clumps. And that can also take place if you have if you're doing a test that's specifically looking for bacteria or anything like that. You can also have that same agglutination take place. So those precipitation reactions that involve diffusion of soluble antigen and antibody can actually be measured using light refraction with a nephelometer. So flocculation testing actually uses the principle of those soluble antigen and antibodies with that binding, which will actually form a precipitate of fine particles. And so what that will actually do is kind of like what we just saw on that previous slide, where you have that tube and you have that flocculation of those particles that just kind of come together in the center to form that little area of precipitation. And there are a couple of different forms of that test method, and we'll discuss that here in a couple of slides. Some of the older precipitation reaction tests that used to be performed in jail are not as commonly performed in the clinical lab today. You may still see some of these test methods in some labs, or you may find them now just more to your reference labs. So most of what you may actually see for this particular method would be using gel electrophoresis. Now there are a couple of older methods, the radial immunodiffusion as well as the Octoloni methods that are often used that use that gel component. We're not going to go a lot into these, just know that these are some of the older methods that actually use that agar, and what it's basically doing is looking for that antigen antibody complex to kind of diffuse throughout that gel media, and so that's basically what you're looking for, is those zones, and you would measure those zones actually looking for that reaction taking place in that gel agar. Your textbook does have this great chart that lists these unlabeled immune reactions and the techniques that we would use to measure them. So it kind of just breaks it down of the technique and the principle of that technique. And we're going to talk about these over the next few lectures. So just refer to this chart when you're just kind of trying to see the differences between them and understand the difference in the principle of these reactions. So occasionally we have to introduce some sort of artificial carrier particles to our test method in order to actually be able to visually see those antigen antibody agglutination reactions. So examples of this would be something like latex particles or a form of charcoal that is used, as well as like erythrocytes. And so your textbook does have a nice little chart to it that kind of gives you some examples of this. And so if you're currently working in a lab, you may have seen these tests before, something like a slide test for C-reactive protein or rheumatoid factor. So like your slide CRP test, using latex particles, it's basically those latex particles have that IgG fraction of that anti-human CRP on it. So you can actually see those particles coming together when that agglutination starts taking place. With the rheumatoid factor, it uses sheep blood erythrocytes that have actually been sensitized with rabbit gamma globulin. And then when you do that mix and you get those reactions taking place, then you actually see the agglutination from those red cells that are taking place. Some of these kits, they use different things. Some of them may use charcoal, some of them may use those latex particles. So I've seen those latex particles in blue, I've seen them in yellow, I've seen them in white. So you get a variety of manufacturers that use different things, but basically it's using those particles to kind of help enhance that visualization of that reaction taking place. There are also different techniques that are out there that are used with some of these different test kits and different test methods. So some examples of them would be like in a direct agglutination, in which you're directly trying to detect the particular antigen that's present or antibody that's present. So an example of this would be a rapid strep kit, where you're actually looking for the presence of that streptococcus antigen in the patient. So if you suspect a patient has strep throat, you do the swab and you do the test. That's what you're looking for. You're looking for that bacterial antigen to be present in that sample. Some of your other methods, you'll learn a little bit more about these as we kind of progress through this course, as well as when we get into our immunohematology blood bank course, especially with these test methods here, when we're doing a direct antiglobulin test versus an indirect antiglobulin test. And so you're looking for in each of these something specific. So your DAT, you are looking for specific antibodies that are actually coating the patient's red cells, whereas in our indirect, we're looking for the presence of particular antibodies that are capable of actually binding some kind of red cell. All right, so to further build upon that latex agglutination type theory, so in these procedures, you actually have antibody molecules that can be bound to the surface of the latex beads. Now, many of the antibody molecules can be bound to each latex particle, which actually increases the potential number of exposed antigen binding sites. Now, if an antigen is present in the test specimen, like the C-reactive protein example that we saw, then the antigen will actually bind to the combining sites of that particular antibody that's exposed on the surface of those latex beads, which will actually form a visible cross-linking aggregation of those latex beads and those antigens. Now, in some procedures, you may actually have those latex particles to be coated with an antigen, so that if they're exposed to the antibodies present in the serum, then you again are going to get that that aggregation of those antigen antibodies, and you're going to start seeing those visible clumps. Now, either any of these methods, especially your latex agglutination methods, usually you can do qualitative test or you can do semi-quantitative. So with your qualitative test, again, it's got that L in there. So it's a letter, positive or negative, versus semi-quantitative, and this is where you would use some of that serial dilution. And so with the semi-quantitative, it's actual numbers. So you're looking at what point is our last positive reaction taking place. So this will be maybe where you would report out that rheumatoid factor as a 1 to 8. Once you reach your 1 to 8 dilution, that was your last positive. Now, co-agglutination and lysosome enhanced testing are variations of latex agglutination. With your co-agglutination, this uses antibodies that are bound to a particle in order to enhance the visibility of the agglutination. It is a highly specific method, but it may not be as sensitive as the latex agglutination for actually detecting small quantities of antigen. Now, one of the older original tests for pregnancy test actually used a form of latex agglutination, and so the rapid, it actually would allow for rapid detection. It was developed originally back in around 1960s, and before that time they were actually using rabbits to determine pregnancy. The facility that I first worked at actually, the older techs would tell me about how they used to have to actually tend to and care for the rabbits, and you know, they always picked that the rabbit died, but actually the rabbit always died, because basically what they did is they inoculated the rabbit, and then they had to kill the rabbit to look for a particular reaction that took place to see if the patient was pregnant or not pregnant. And of course it was based off of some of those pregnancy type of hormones. But basically this older method was based on the rapid detection of direct monoclonal latex slide agglutination, in which they were looking for the HCG to agglutinate with a latex particle that was actually coated with an anti-HCG antibody, and if it was to look to see if it was present in that test. So obviously this method is not really used that much anymore. Pretty much now you're going to have the typical pregnancy test that we think of as far as that lateral flow, or you're going to do the testing on your chemistry analyzers. And I'm sure once you get to clinical, you will get the opportunity to actually do a few of these latex agglutination type tests. Some of the more common ones that you're probably going to see would be C-reactive protein, rheumatoid factor, rubella antibody. But basically what you're going to see is that you have some latex beads, and for your latex agglutination test, you've got latex beads, and then you also have a particular antibody. Those beads are going to get coated by the antibody, and then it's going to agglutinate. That antibody is going to agglutinate with an antigen, and basically those latex beads are going to be what will actually help to visualize any of that agglutination that's taken place. You also, another one of the agglutination methods, you can see where you have the lysosome. here that's present and that has those particular antigens on the site, and then you're looking to bind that with the latex bead. There are several things that can cause false positive and false negative reactions for agglutination tests. We're really going to kind of dive a little bit deeper with this once we get into our immunohematology blood bank course. You know, of course, some of the more obvious things will be like not adding your reagents, not properly centrifuging, not properly allowing the appropriate amount of time for the test reaction to actually take place, allowing it to sit too long, or not reading it soon enough, as well as things like something that we call a prozone effect. So kind of review over these and just see what the outcome is. And then as we get into our immunohematology course, we're going to kind of talk about these things a little bit more in depth. So there are some alternative type procedures that are used. One that we've already mentioned was that flocculation test. So these tests are based on the interaction of soluble antigens with antibodies, and you're looking for that precipitate of those fine particles. You can look at those macroscopically or microscopically, just depending on what the particular test method is, and that's because you're going to kind of keep it to a confined space. So this is going to be more like those slide agglutination tests. Some examples would be like doing a VDRL or an RPR test looking for syphilis. You also have direct bacterial agglutination, which is actually used to detect antibodies directed against certain pathogens. The most basic test will actually measure the antibody that's produced by the host to the particular antigen determinant that's on the surface of that bacterial agent in response to an infection because of that particular agent. Hemagglutination. So this particular method of testing detects antibodies to erythrocyte antigens. So the antibody containing specimen can actually be serially diluted, so we can do those dilutions in the series, and then a suspension of those red cells are actually added to the dilution. If you have a sufficient concentration of the antibody present, then those erythrocytes are cross-linked and they will actually agglutinate. If non-reactant antibodies or an insufficient amount of antibodies is present, then those erythrocytes will not agglutinate. By binding the different antigens to the red blood cell surfaces in indirect hemagglutination or passive hemagglutination, the actual hemagglutination technique can be extended to detect antibodies to antigen other than those that are actually present on the red cells. Now, occasionally we might have to have some sort of chemical agent added in order to kind of enhance or help those bonding bridges and those reactions to take place. Some antibodies, for example, IgG, do not directly agglutinate erythrocytes. So this is like an incomplete or a blocking type of antibody that can be detected using an enhancement media such as anti-human globulin. And so that particular reagent, it was actually once referred to as Coombs, but that was just the name of the person that invented the test. It's not used today. You will hear some physicians still use it, some of your older physicians, but as my instructor told me, don't use that word because Coombs is a dead man now. So basically, once you add that AHG reagent to your reaction, then it's basically like a second antibody, and it's actually going to bind to the antibody that's going to be present on those erythrocytes. And we'll talk about this in more detail as we kind of progress through this course, as well as go over to blood bank. But your diagram here at the bottom is showing you those agglutination reactions where you have the red cells and we have the antibodies, and then those come together and start bridging together to form those clumps. And then you also have two bottles of reagent. One is anti-IgG, which is a monoclonal. It's just the one immunoglobulin that's present in that sera. And then you also have anti-IgG C3b, C3d, And this second reagent is called a polyspecific reagent because it has more than one of those more than one of those immunoglobulins present in that sample. Actually, it's not just immunoglobulin, you have your complement in there as well. So now let's talk a little bit about the mechanisms of agglutination so that we can kind of understand this and why we do some of the things that we do when we do all of this different testing. So in a nutshell, agglutination is basically the clumping of particles that have antigens on their surface, such as erythrocytes, by an antibody molecule that will actually start forming those bridges between those antigenic determinants. Now this is basically that endpoint reaction that we're looking for with most of our tests that are involving any sort of erythrocyte antigen. Agglutination is influenced by a number of factors, and it is thought to happen in two stages. So your stages of agglutination are sensitization as well as a lattice formation. Now we're going to talk about each of these. So the first phase of agglutination with your sensitization, this is the physical attraction of the antibody molecule to the antigens that are actually present on that red blood cell membrane. So the combination of antigen and antibodies is actually reversible. It's a reversible chemical type of reaction that takes place. So once it happens, once you get that agglutination, you can actually reverse it. You can break those bonds and those bridges. So we can actually do this by altering the physical conditions that are present. So if we forcibly, purposely manipulate those physical conditions, then we can actually break that antigen-antibody complex. And then basically what that's going to do is release that antibody into whatever it's in. And releasing that antibody is what we refer to as an elution. So there are different procedures out there. The most common one, as far as where you'll have this referred to, is going to be within blood bank. But basically, an elution is basically taking and trying to extract an antibody from something. Now the amount of antibody that will actually react is affected by the equilibrium constant or the affinity constant of that particular antibody. So in most cases, the higher the equilibrium constant, the higher the rate of association and the slower rate of dissociation of those particular antibody molecules. Now the degree of association between antigen and antibody is affected by a variety of factors, and so we can actually alter those in many of the cases in vitro or in a test tube by actually altering those factors that influence that antigen-antibody association. So those factors are listed here. We're going to kind of go over those in a little bit more detail. But first, let's just talk about lattice formation. So lattice formation, or the establishment of cross links between sensitized particles, in this example your erythrocytes, and antibodies, will actually result in an aggregation. And of course this is a much slower process than the actual sensitization part. You know, the coding of those antibodies and everything that takes place, that's pretty quick, but actually starting to bridge those bonds together because of that environment that it's in is where this is a slower process. Now the formation of those chemical bonds and that end result of that lattice formation is dependent on the ability of a cell with that attached antibody on its surface to actually come close enough to another cell to actually allow that antibody molecule to start to bridge those gaps and then come together to form ultimately that larger molecule of those cells clumped together that we're looking for as far as that reaction. Now as those antigens and antibodies actually start to combine, then you're going to get a multi-molecule lattice that will actually start to increase in size as it precipitates out of the solution as a solid particle. Now cross-linking is actually influenced by various factors, and one of those is zeta potential. So inert particles like latex, red blood cells, bacteria, they all have a net negative charge on the outside surface of their cell. And this is what we refer to as the zeta potential. So you can see here in this depiction where it kind of shows you got the surface of whatever it is, and then you kind of got that ionic cloud around the outside of it. So that negative charge is actually kind of pushing away at other cells or organisms or particles or whatever is around it. Now, if we actually alter a little bit of the medium that it's in, then we can kind of help to reduce that negative surface charge and bring those things a little bit closer together. And we do this by what we call reducing the ionic strength of the reaction medium. And what this will do actually allow that antibody to uptake to that cell a little bit quicker. Now, charges can be overcome by a couple of different methods, including centrifuging. This is kind of like in blood bank where you put those antigens and antibodies in the tube together, spin it down really fast, and you force them to come together quickly. We can also do the addition of charged molecules or enzyme pretreatments in order to permit that cross-linking. We can add things called low ionic strength solution, so that kind of helps with reducing those charges within that particular medium. The pH of the solution will actually come into play as well. So if the pH of the medium that's used should be at or near the physiological condition or an optimal pH, which is like 6.5 to 7.5. When we have that neutral pH, then the high electrolyte concentrations will actually act to neutralize that net negative charge of those particles. Reactions can also be temperature dependent. Your IgM type of antibodies, they react better at colder temperatures, whereas your IgG reacts better at your warmer temperatures. Kind of a good way to kind of think about this is your IgM, of course, are your ones that are going to respond first. They're quick, they're first out of the gates, they don't even have time to grab their coat. So obviously if they go out there in the cold, they're gonna do the job faster because they're ready to get back inside. Your IgG, they're the ones that they're last to react, they're taking a little bit longer, they like that cold, that warm temperature, so they're not going to go out in the cold right away. They're going to be bundled up, so that means nothing can attach to them quickly, so you're going to need that anti-human globulin to actually show that those IgG antibodies are present. The duration of time that you allow this reaction to take place, that can have a factor on it. Most of your tests that you're going to do are going to be anywhere from 15 minutes to an hour. There are some tests now with the development of the reagents to be a little bit better. I have seen some tests that are as quick as 5-minute incubation times. All right, so let's talk some more about some of those other mechanisms. So antibody type. Our IgM molecule, as you recall, is pretty big. You've got several of those binding sites that are available in that one IgM molecule. Our IgG, as you remember, is just a Y, so that's a little bit smaller. So it's so small, it really can't overcome that electrostatic force that's happening between those cells. It can't overcome that zeta potential. So what we have to do is actually use anti-human globulin to cross-link those bridges between those antibody molecules in order to be able to have that visual appearance of an antigen-antibody reaction. Now, another thing that we have to note is the antigen-antibody ratio. So ultimately, we want it pretty equal, the amount of antigen binding sites as well as the antibody that's available, in order to have that reaction take place at what we call a zone of equivalence. So that's basically, you're almost 50-50. You got enough antigen and you got enough antibody to create that reaction. Now, what can happen is we can have something called prozone or postzone. So with your prozone, if you look up here in this top corner, what this picture is, so for your prozone effect, you're over here to this side. If you have an excessive antibody concentration then what's going to happen is you're going to have what's called a prozone phenomena, and it's going to result in a false negative reaction. So you've got too much antibody. Those antibodies aren't able to come together and actually form those bonds because the whole cell itself, let's just say you only have 2 antigen binding sites, because ultimately you need at least 2 so that you can have connections. What's happening is that antibody is just sitting here binding, and then it's never bridging any of those gaps. So you don't have any of that lattice formation that's taking place because you got too much antibody and not enough antigen. The opposite is going to be the post-zone effect. So in your post-zone effect, you have too much antigen, and then you don't have enough antibody to actually help to form those bridges. And so this is what we call post-zone phenomena. And so you might actually have some small aggregates or clumps that could be visible, but not always. But basically you're not really having much of that lattice formation to give you that good solid clump to take place. Antigenic determinants. So the placement and number of the different antigenic determinants can actually affect your agglutination reaction. As I just previously mentioned, if you've got a cell that only has 2 antigens on the surface of it, you know, you don't have as many of those bonding spots. It's going to be a little bit harder to start bridging those bonds. You could have that antigen get bound by that antibody, but then it doesn't have another antigen receptor to actually finish making those bridges. You could actually have some situations too where those particular sites are just really small, or they're kind of buried deeply into that cell membrane, so those antibodies aren't actually able to make contact or good physical contact with those antigenic binding sites. Steric hindrance is an important physiochemical effect that influences the antibody uptake of the cell surface antigens. So what happens here is you can have some dissimilar antibodies that will that are basically approximately the same binding constant, and what they will do is they'll kind of be directed against those antigenic determinants that are located on those cells, and so the antibodies will actually start to compete for space, trying to make that reaction take place for those specific sites. So they're basically just fighting over who's going to bind what first. But in addition to that antibody competition that's taking place, you can also have some things like complement, other protein molecules, different things that can actually be affecting the surface of that cell, so it can actually produce that steric hindrance as well. Agglutination reactions are graded, so the degree to which we see that agglutination happen is what we scored at. So sort of like in hematology, as well as urinalysis, where we kind of had to grade the morphology of the cells, the red cells, as well as the amount of something that was present in the urine. We do the same thing when we're grading these agglutination reactions. And so we're going to kind of walk through them just kind of briefly, but I'm not going to go into a whole lot of detail because, again, once we hit blood bank, we're going to focus on this a lot. So you will become very familiar with these reactions and what they mean. A negative reaction, of course, is just you have no agglutination taking place. Mixed field reactions means you have a mix between a positive and a negative reaction. A weak reaction is just going to be that you have some very tiny stuff that's happening. A lot of times you have to kind of refer to the microscope to confirm that you're actually seeing these reactions take place. You've got 1+, 2+, 3+, and 4+ reactions. And the 1+, the lower end, is just basically you've got some very small clumps that are taking place all the way up to one huge large solid clump. So here again we kind of got that divided out. The only thing is the mixed field is not included on this chart. But again, basically you've got your negative, there's no agglutination, your weak reactions. A lot of people will refer to this as microscopic. You can usually tell a little bit is happening if you look at it in the tube, but then once you go under the scope, you start seeing those aggregates that are taking place up under the scope. You can see those little clumps that are happening. With your 1 Plus, you've just got kind of like some hazy cells in the background that don't really look like they've agglutinated, with some very small clumps. And then as you move up to the 2 Plus, the clumps get a little bit larger. 3 Plus, you have a lot of large clumps, then some small ones, and then your 4 Plus is just one solid button in that tube. Now, in immunohematology, there is a gel matrix that is actually used. So what we actually have here in the lower part of this sample well is the actual gel beads that are in here. So they're kind of a clear, hazy looking bead when they're all in there. And so these beads are just kind of lined up. They're stacked in the bottom of that tube. So what will actually take place is you will place your patient's serum or plasma into the tube, patient's plasma, as well as red cells, just depending on if we're using the test red cells or the test reagent, whatever we have to put in this well. But basically, everything's going to kind of go in here, our antibodies as well as our red cells, whether it's patient or a test reagent. It's going to go into that reaction well. We're going to allow some incubation time, a centrifuge. And so we're basically allowing time for those red cells to start to come together and forming those bridges from those antibodies that are going to kind of bridge them together. Now, the red blood cells themselves, when they originally start off, are a little bit smaller than these gel particles. And so what will happen is if you have a negative reaction, it will flow through that gel well and then sink all the way to the bottom. So that gives us our negative reaction. However, if we have agglutination that takes place, you can obviously see that this whole entire molecule here will not go through that gel. So when it's spun, it's not able to be forced down through those gel particles. So you actually get that visual line across the top. If you just have a few agglutinations here and there, so some of those cells may be able to actually make their way through that gel, just depending on how large those clumps are, how much antibodies present, whether or not you had that prozone effect, variety of things. But at least with the prozone effect, sometimes you still can see a few little aggregates take place. But basically, this is just kind of the overall concept of that gel method. And we will learn a whole lot more about this once we get into blood bank. All right, let's talk about microplate agglutination reactions. So you can use microplates in a couple of areas of the lab. Primarily, it's going to be blood bank and micro where you're going to really see these types of testing going on. With your microplate reactions, depending on the particular test, you could potentially, within those little micro wells, so you're going to have a plate that is going to have several of these wells in it. And so basically you would have this portion here within this little microplate. So you're just going to have all of these little wells. And then you're going to add a very minute amount. So the fact that it says it's a microplate means you're using a very small amount of reagent and sample. Now in the bottom of each of those wells, depending on the particular test that you're doing, some of these may actually be coated with something around the edges of this well. So once you put the reagent and everything in here, allow it the time to react, then what you actually have taken place is you have binding to the side of the well. Whereas if there is nothing there to actually bind, then everything will just kind of just flow down until it kind of comes to a stop here at the bottom and kind of all piles up together. So what you're actually looking for is the opposite of the tube, whereas the tube, our positive reaction was that solid little button. It's the opposite here, where you actually have everything that's kind of stuck to the bottom or outside edges of that well. So this is considered your positive, and this is your negative. So in this particular test here, they did a dilution of some sorts, and they had positive reactivity all the way up to the 1 to 160. So everything after this is all considered negative. This is all positive. So this, the last result of the positive, is the part that we would actually report out. So blood bank, you're going to see a lot more of the blood cell looking agglutination. Some of your tests in micro, if it's using bacterial particles or whatever is being tested for, then you're going to see something more like this, where you're just looking for that haze of where it's attached, versus this where you've got that one little button that's been kind of formed in the center of it. Wow, we've come a long way with those precipitation agglutination reactions to get back to precipitation reactions. So your precipitation reaction, as we previously mentioned lectures ago, is done by doing nephelometry. So the quantity of cloudiness or turbidity in a solution can be measured using a photometric type of method. So when you have specific antigen-coated latex particles acting as a reaction intensifier, then the when they actually agglutinate with that corresponding antibody, then what you will have is an increase in light scatter of that solution, and that's what we actually are going to measure when we're doing nephelometry, to actually look for that macromolecule complex. Now in immunology, we actually use it to measure complement components, immune complexes, and the presence of a variety of different antibodies. And of course, nephelometry is based on the principle of light scattered by a homogeneous particulate solution at a variety of angles. So there are 3 types of scatter. You can have scatter around the particles, you can have forward scatter caused by out-of-phase backscatter, and you can also have forward scatter exceeding that backscatter. So nephelometry does use the Heide-Ledberger curve, and so this should look familiar because we just kind of talked a little bit about this. Where basically if you have antibodies that are present in excess, a proportional relationship exists between the antigen and resulting signal. If the antigen overwhelms the quantity of the antibody, then the measured signal drops. So it's basically that pro-zone and post-zone effect that's taking place there. All right, so with nephelometry, then what's actually going to happen is you're going to have a specific polyclonal antibody that's going to be added to a cuvette along with the test sample that has the particular analyte that you're looking for. You're going to allow that reaction to take place, whatever the particular procedure is, and then you're going to have some sort of light source. It's usually going to be some sort of infrared LED type of light source that will be used, and then you're measuring that light scatter that's actually going to happen with that reaction. There are some disadvantages to this particular test method, one being that it has a very high initial cost to set it up, and the other is that there are interfering substances like microbial contamination that can actually cause a protein denaturing, and then that's going to give you some erroneous results. The newer methods actually allow for testing with a cold treatment that will actually look for and analyze cryoglobulins. So cryoglobulins are actually proteins that will precipitate or gel when they're exposed to colder temperatures, but they can be dissolved when they're heated. So one of the phenomena that you might recognize is the Raynaud's phenomena, where people will get that bluish decoloration, bluish to white in their fingertips and finger pads. And it's just got to do with those cryoglobulins that are within the patient. So those extremities aren't, the temperature in the extremities aren't as well maintained, so you can see the visual appearance of that taking place.